I'm Meenakshi Sharma, working as an assistant professor in ITS Pharmacy College. I would like to share my presentation about plant tissue culture and their advantages. The learning outcomes of the plant tissue cultures, the learner will able to understand what is plant tissue culture, the history behind the plant tissue culture, and advantages of plant tissue cultures. About the introduction compiles the definition of plant tissue culture is a technique of growing plant cells, tissues, organ, seed or other plant parts in a sterile environment on nutrient media. We can also define as the plant tissue culture is in vitro condition of plant cells or tissue under aseptic or controlled environment, and controlled environment means the inside the labs with having the controlled atmosphere in a liquid, or we can say the semi solid, well defined nutrient media for the production of primary and secondary metabolites or to regenerate the plant. In vitro conditions means inside the lab conditions. So we can process the method inside the lab which are having the very controlled environment. Or in other words, we can also explain experimental technique through which a mass of cells, that is callus, is produced from the explant tissue. The explant means that is a part of plant which are used for the cultivation. The different explants which are using for the cultivation methods. Here, in field cultivation, we use the stem part or we can use the seed part. But here in plant tissue culture, we can use the different part of the plant known as explant. Let's move to the history. History confined that is establishment of the plant tissue culture. Like in 1902, the idea of totipotency of plant cells was given by Hublet. That is, the totipotency means that the capacity to grow of cells in the media. In 1937, White first time established successful the root culture of tomato. That is, here we are using the explant as root. In 1941, Vancouver used coconut milk for the growth and development of young tetra embryos. So here we change the part of the media. The coconut milk is used for the further growth. In 1957, Skoog and Miller demonstrated the role of auxins and cytokinins on the root and shoots formation of tobacco tissues. Like auxins and cytokinins are the growth hormones which is responsible to uh, their growth for further issues. In 1962, the Mushin and Miller introduced the medium for the tobacco culture. Further, in 1987, the isolation of gene from the bacterium Bacillus in the Frankincense. Like furthermore, establishments of the plant tissue culture can be held up. The reflection spot. Reflection spot means why we do need of plant tissue culture as we can do by simple cultivation method. Every plant can be cultivated through the field method, that is a cultivation method. Why we need that? So there are so many advantages over for that cultivation techniques as we discussed in this class. Next slides. Like advantages of tissue culture. Like first advantages, that is availability of raw material. With the discussion of availability of raw materials means some plants very much difficult to cultivate and they are also not available in abundance. Abundance means everywhere. And tissue culture technique is considered a better source for regular and uniform supply of raw material for medicinal plant industries for production of phytopharmaceuticals. So we can overcome this problem through this plant tissue culture. Next is like same like this availability of raw material, that is fluctuation in supplies and quality. Here the not difficult to grow, but the variety of your fruit drugs is variable in quality due to changes in climate. Sometimes climates are good, sometimes bad. So they due to that climate condition, the changes of secondary metabolites may happen. Further, the crop disease and the season also. All these problems can be overcome by tissue culture. Third one, a new method for isolation. So new method for isolation means it is possible to obtain new methods for isolation on newer compounds from plants by this technique and for the which patent rights can be obtained. So in the cultivation method, we cannot apply it for the patent. But here the method validation or method process, we can uh, patent right, or we can also patent for the product also. Next is a biotransformation. Biotransformation is nothing but a process through which the functional group of organic compound are modified by living cells. So here we can change the functional group of the organic compound. Reaction are feasible using plant cells culture. So here we can change the path, we can change the function group of the organic compound. Next, disease free and desired properly. That is a large scale production of the plant with disease free and desired property could be stored and maintained without any damage during the transportation for subsequent plantation. That is all about the disease-free and desired property. Like next is a biosynthetic pathway. It is very important that here we can use or we can choose the tissue culture can be used for tracing the biosynthetic pathway of secondary metabolites using labeled precursor in the culture media. But we cannot apply this method over the cultivation techniques. So here we can use that method for the different changing of the pathways of secondary metabolites. Furthermore, advantages are there, like next is immobilization of cells. Immobilization of cells means that is the cells which are entrapped into the different media for the further use. Or you can say that the tissue culture can be used for the plant preservation by immobilization. Immobilization is nothing but an entrapment of the cell for further facilitating transportation and biotransformation. Here one of the example we can use that the simple cells can be entrapped into the sodium, 2% of sodium alginate, and we can prepare the beads, and that beads are for use for the further process, and that can be stored for long time also. Next point, that is a continuous uniform biomass is obtained. Like continuous uniform biomass is obtained through the plant tissue culture, and that biomass is a nothing but a different cells which are obtained through the plant tissue culture. Next point, medicinally important compound can be synthesized, which can be synthesized chemically. Medicinal important compounds means like from the molecule or by the changing of the biosynthetic pathway, we can synthesize the different compound from that particular plant tissue culture. Next is useful natural compounds can be processed, and that is interdependent of the soil condition and the change in climatic condition. So having very good nature of the secondary metabolites through the plant tissue culture, and they can be, the secondary metabolites is also obtained through the cultivation method, but here the nature of the natural compound is very good in nature or having very high level of principle in that. Next is improvement of the medicinal plant species. We can improve the different plant species through the plant tissue culture by changing in their pathways. Next and last is propagation of plant without seed in defined and controlled conditions. Because in cultivation method, their propagation done by only by the seed or sometimes by the stem part. So here we can use any type of the plant, any type of the part of the plant. Here we can use it for the plant tissue culture. We can use the shoot tip culture, root tip culture, flower culture, embryo culture, ovule culture. We can propagate through the any part of the plant of for the plant tissue culture. That is the importance of the plant tissue culture, or we can say the advantages of the plant tissue culture. The furthermore required slides for the further information is there, and the advantages is like it can be issued, it can be revived through time to time. And thank you very much for listening my presentation, my slides. Thanks.